Hello, my name is Eric Hollenbaugh,
And I'm Riley Galt.
and today we're going to be talking about the research we performed on Tetrahymena thermophila. And we were looking at the effect of EDTA on CalModulin 1 gene expression and cell viability. So to start, Tetrahymena thermophila are unicellular eukaryotic ciliate. They're very popular as a model organism and used a lot in research. EDTA is ethylene diamine tetraacetic acid, and it's a metal ion chelator that's present in a lot of body cleansing products, so like soaps and shampoos, and it's even present in some fortified foods. Calcium is something that would be sequestered by EDTA, and it's a metal ion that's present throughout the body. CalModulin, short for calcium modulated protein, is an intracellular target that's activated by calcium. And so the CalModulin 1 gene, or just CAM1, codes for this CAM1 protein. And that's needed for many important biomolecular pathways. And so our hypothesis was that if Tetrahymena thermophila are exposed to EDTA, then the CAM1 gene expression will increase. And cell viability, along with the number of feeding vacuoles, will decrease.
Yeah, so now we'll move on to the methods that we used in order to complete this research project. For starters, we had to make primers that were specific to our CAM1 gene. To do that, we utilized a software called OligoAnalyzer. Once we had the primers, we began culturing our Tetrahymena thermophila. Initially, they were placed into NEF media, which is nutrient poor media, and then once experimentation began, they were transferred into SPP media, which is nutrient rich media, and this pretty much allowed for the Tetrahymena to replicate as they normally would. Once that was done, we separated the Tetrahymena into two different flasks. One received the 24-hour dose of 5.0 millimolar EDTA, and the other received the same amount but of water and not EDTA. Once the 24-hour exposure period was complete, we extracted the RNA using Quigen's RNase mini kit. After that, we used reverse transcription, and this was utilized to take that mRNA that was extracted and turn it into cDNA. Once that was completed, we then did a quantitative-PCR using PowerUp CyberMix. To kind of have a baseline level of gene expression, we used a control gene called BTU1. And then finally, during the 24-hour exposure period, we did cell counts of our Tetrahymena every 6 hours using a hemocytometer. And then after each round, after the 24 hours was complete, we did a feeding assay that was performed with India Inc.
Yes, moving on to our results. If you look at figure 1, we just put in a chemical structure of what we used, which was tetrasodium EDTA, which we just dissolved down in water and made it 5.0 millimolar. Figure 2 is an image of the Tetrahymena after they had been feeding on the India Inc. dyes during the feeding assays, and you can see the little vacuoles there in the 5 cells. And then figure 3 shows the average number of feeding vacuoles. And so there's quite a statistically significant difference there, even if you're just looking at the data. But then we also did a t-test for that, and the p-value was significantly low, below 0.05. And there were 3 replicates that we did for that. Figure 4 is the average number of cells per milliliter of media, and so that was the cell counts that we did using the hemocytometer, and there was quite a significant difference between hour 12, hour 18, hour 24, but in the statistics of it, there was only a difference between hour 18 and hour 24 for those 4 groups. And then figure 5, it shows the fold change in expression of CAM1 after we ran the PCR. And again, there appears to be quite a significant difference there between the means, but we weren't able to run another replicate of that, so we couldn't be for sure if that would be how it turned out every single time.
Yeah, so to conclude for the research, we will start with the feeding vacuoles. So the number of feeding vacuoles was significantly increased in the controlled treated tetrahymena, as you can see, and as Eric mentioned, the p-value was below 0.05, indicating a significant difference. Then moving on to the cell counts, there was only a significant difference in the 18 and 24-hour mark, where the Tetrahymena thermophila were more abundant in the control. In the 6-hour and the 12-hour, there was not significant results found. However, it was still following the trend like seen in 18 and in the 18-hour and the 24-hour cell counts. Further, for our single round of qPCR, it could not be statistically analyzed because only one round was completed, but it did show promising results. As you can see in figure 5, there is a big difference in the fold change in expression of CAM1 in the EDTA treated Tetrahymena thermophila. So overall, our results do agree with our hypothesis regarding the number of feeding vacuoles and cell growth as far as being significant. In addition, the appearance of increased gene expression further supports our hypothesis. Although it was not significant, it is definitely a starting point where future research could begin. And in order to get better data, replicating this research would be beneficial.
Lastly, we'd just like to throw in some shout outs, some acknowledgements to our great faculty advisor, Dr. Stephanie Otto-Hitt, for her superlative guidance, enduring patience, and unwavering support. And that is all. Thank you for joining us.
Thank you.